Nice catch. Okay, guys, I think we're back in business. I ended up opening another PowerPoint. So if you are not seeing this at home, let me advance it. Quickly let me know. Hopefully you're seeing a picture with cells. If that's not the case, pipe up. So we've got a handful, quite a big handful, of slides to cover. I expect getting about halfway through this and then wrapping up the rest of it on Friday. The test will open, I think I've got it set to open at 10 o'clock on Friday, if I remember right. It is timed. I've set the time for 2 hours. It's open book, open note. So if you've got questions or concerns, let me know beforehand. It will be posted on Moodle, so you take it directly from there. Yes.
How long is that available?
It opens Friday and it closes Monday at 5 p.m. Okay, other questions about that before I jump into this? I will tell you right now, I have not, all I have done, you might see it on Moodle. I assume you potentially see it. There is nothing there. It just says the exam. I have not populated it with questions, so I can't give you details, but that's gonna happen. And Friday, I will give you some of that. Yes.
So is it automatically graded like after we're taking, when we get our grades, or do you separately look at it?
It will be automatically graded, but you will not see what you got until after 5 p.m. on Monday. Okay. Other questions? All right. So this chapter has a lot of stuff we want to think about as we move through here and start thinking about microbes. We'll want to show you a little bit of detail about them, not that we haven't already done some of that. We have talked about gram staining. Real quick, is there a study guide? I do not think I'm going to get a study guide posted. It's open book, open note. So We will see. I will tell you that it is probably a good idea to read the end of the chapter summaries. Real quick, you can go through that stuff and get an overview of what that chapter was about. And then if you feel like there's some fuzzy area, you can go back and kind of review that. Okay. Is that my same chat? Okay. In this classroom, that's all fine and good. And I could see if people are doing that. When I'm teaching in the distance room, it's still over Zoom, but I don't see any of that stuff. So I'll try to pay attention to that. Okay. So there are a lot of concepts that kind of get thrown out here as we are looking at kind of this overview of cells. We have mentioned stuff like gram-positive and gram-negative cells. We've talked a little bit about that, and I showed you some gram stain images. We're going to give you some details as we move through and think about cells as far as why a positive is positive, as we look at some zoom in and see some features of the cell, and conversely, what makes a gram-negative negative, and how those stains, although we showed you those images, how do they actually work, right? So this is a smattering of all sorts of organisms. We're not going to get bogged down with all of the details and describe what everything is. I could tell you there are a number on here that I'm like, oh man, I could talk all day. I could talk all day about Giardia right here, or maybe some of these trypanosomes. These are both eukaryotic cells that are parasites, and we will see some details with them a little bit later. So as we get started with this, we need to kind of back it up and think about some ideas that set the stage to really kind of think about where life comes from and how to approach that idea with the scientific method. So the thinking was that organisms show up spontaneously. As we look at something like this meat that gets maggots on it, we take lots of stuff for granted today. You go to the supermarket, all of the meat is there in the refrigerator or freezer section, but in the refrigerator section, you can see all sorts of stuff. It's kept at a nice cold temperature to prevent it from spoiling, right? But back in the day, that wasn't the case. And you would go to the market and here would be this meat and you would take it and you would go home. And if you don't consume it rapidly, well, you're going to be in trouble, right? That stuff's going to foul, right? It's going to get organisms like maggots on it. And then we start thinking ice boxes and then refrigeration, you know, all of these things. We're not concerned about that, all right? The question, thinking back in the day, pre-refrigeration ideas, how in the heck did these maggots get all over my meat? I just bought this a week ago, right? Or I just killed this animal a week ago, right? How is it that I'm seeing all of these things? It must be spontaneous generation, right? We know, or we should know, that these maggots are the larval form of flies. As the fly lays its eggs, they hatch out and start feeding on these tissues and then develop into the fly, right? We know that now, but back in the day, we did, right? And it was pretty easy to write that off as, maybe not pretty easy, but that was the way we answered these things. It must be spontaneously. Through the abyss, these things just showed up, right? There were numerous people that started questioning that and thinking, you know, maybe there's something more to it. Maybe it's not just spontaneous. So Francisco Reedy thought he could set up this experiment, and really we're looking at a couple of experiments all shown on the same slide. You technically need to kind of disregard the gauze covered beaker here for a second, all right? His initial experiment was to take two pieces of meat, one of them left open and the other corked up so it's sealed. No flies can get in there, right? As this happens and he lets it sit for a couple of days, you can see the maggots in here and nothing over here, all right? I bet you, if we were to look a little more closely, we could notice some other stuff going on, like maybe some bacteria, the meat starting to get, putrefied. There's probably some other stuff going on. We'll get to that in a little bit, right? Where the heck did that come from, right? So the maggots show up here, but not there. And he thinks, done deal, right? It's not spontaneous. It's gotta be from the fly. The fly is what's bringing that in there, right? That seems to make sense, right? Like I said, we're really looking at a couple of experiments up here. And this whole idea was spontaneous generation. It took centuries, which seems crazy to think about, but it took centuries to truly go through these processes and multiple types of experiments that we're going to show you as we move through here to truly get rid of this idea of spontaneous generation, right? That was how we thought, right? So the naysayers are like, oh, well, the issue is you corked that sucker up and there's no way air can get in there. And the air is what we need, like this whole miasma idea, right? The foul air must be where all of this nastiness is coming from. If you don't give it exposure to air, it makes perfect sense why there's nothing growing on there. The air is where it's coming from, right? Spontaneously through the abyss. So he's like, okay, we'll set up another experiment. We put some gauze or some cheesecloth over the top of his flask, redoes this experiment, and as we can see in this image, there are no maggots on the meat, but up on the top, on the gauze, where the flies landed and are like, it smells delicious in here. I should be laying my eggs. That's where they do it. And the maggots show up on the top of the cheesecloth. So again, Reedy thinks, sweet, I have done this. I have just disproved spontaneous generation, right? It's obvious that these maggots are not spontaneously evolving on this rotting flesh. They got there from these flies, right? It's not so simple, right? And as I said, what about the nastiness on the meat? Not the maggots. What's happening with that, right? Well, maybe bigger forms of life, maybe that makes sense. But what about this other stuff, right? We can't understand that at all. And that obviously is spontaneous generation. That's how it gets all rancid and putrefied, right? So we will transition here in a second, all right? So Reedy demonstrated that, and I love these guys. I think it's awesome to see some of these images. So he shows the maggots were offspring of flies. It's not spontaneous, all right? Needham over here, he argues that the microbes, the sliminess on the meat, well, that crap must be spontaneous, all right? That's got to be some life force that's generating it, right? Not, as we know, bacteria that's already there. Spallanzani, and these guys are all kind of, like I said, this takes a long time as we're progressing through. Spallanzani shows up and he's like, I think we can further figure this out. And he goes through and tries to disprove these ideas that Needham is throwing out there. And again, the miasma, all of this stuff is still feeding all of these people. We shift ahead a bit to our good friend Pasteur. We say he's the one that truly put the nail in the coffin with spontaneous generation, but that's not 100%. All right, and I think I have an image, but we'll talk about the next one here in just 1 second. All right. Excuse me. So Pasteur, he did all sorts of things. All right, and we think of kind of a father of microbiology. He did more than pasteurize your milk. All right, lots of stuff we credit Pasteur with, all right? He did numerous things, and one of them is proving spontaneous generation, right? So these flasks, these are called swan neck flasks. These are flasks that he built. He took a flask, he heated it in his flame and bent the glass into this curve, kind of like the, the trap under your sink, right? To keep the sewer gas smell away, right? This little trap's got a bit of water in there and you don't smell that foul odor, right? It's kind of the same idea like that, right? This is kind of like a little trap, right? There's not water or liquid in there, right? It allows the air to get in, but it's still kind of a trap, preventing stuff like dust or microbes that are possibly floating around in the air. Some of you guys, when you did your first lab and you were looking at things, maybe left the lid off of the Petri dish and just let it sit. I know the manual mentions you could do something like that. We kind of turned it into just a free-for-all. We're like, whatever, swap what you want. Let's see what grows, right? I feel like that's a little more fun, all right? But taking a Petri dish and just leaving it exposed to the air for a half an hour or something, and it could be, we've done things in the past where the TA does it right on the front of the bench and they give their lecture. This is pre-mask time, right? They give their lecture after a half hour of talking and doing stuff and people walking around, they put the cap on it and incubate it up. And we see all sorts of things, right? And you're like, oh, is that spewing off of that person? Maybe, right? There are others where maybe you put the Petri dish clear over away from where anybody's going to be, right? And you leave the lid off and you find there's still massive amounts of stuff on there, all right? So we know now there's all sorts of organisms floating around in the air, right? We know that now, right? So the idea with this and what he is working with as we look at this water, it's a type of broth, right? Bacteria, organisms like nutrients, right? It doesn't just have to be bacteria. When you put your bacteria or you swab the environment, you put it on a plate, that plate has nutrients within it, right? You got to give them some sort of food, right? Like that slab of meat we saw with the maggots, feeding on, right? So these are broth mixtures that he produces, and they are meat broths, meat infusions, right? So he takes something and cooks it up, right? And gets all of this broth, and there's lots of good stuff in there that things could potentially grow in. All right. So he heats it up and basically cooks it and sterilizes it. Anything that was possibly in there, we would say is dead, right? He boiled the crap out of it. He lets the flask set for a little bit of time and it stays the same color. It's basically kind of clear. There's no sorts of organisms that were growing in it. All right. So that seems to fit with this idea. All right. He then does another one and he breaks, multiple ones. He's got one that he's cooked and never did anything with. He cooks this one up, but he breaks the top of that off and lets it be exposed to the air, right? As these are both sitting out on the bench top for a week or whatever, this one stays clear and this one gets turbid. It gets all kind of cloudy, right? You can't see through it because there's all sorts of stuff growing in there, right? Done deal, right? This stuff is coming from microbes, right? There are things in the air that's causing this to happen. There's fungi, there's bacteria, there's all sorts of things floating around that ultimately caused this to foul, if you will, to get all contaminated, right? He did other experiments with this, with these swan neck flasks. He would do this same thing, heat it up, and then after this flask up above has sat for a week or better, he could take the flask and tilt it so that the liquid goes up into the neck, touches the dust and stuff that's settled in there, that's got microbes on it. As the liquid touches it and then comes back into the flask, it also goes turbid, showing that it is stuff that was in the air that settled into there. And that's where all of this stuff is coming from. Not spontaneously. Yeah.
Okay, so that's kind of what I was leading into. Like, how is he able to take the population and go, oh, it's not just the air, because based on that, society could probably go, oh, it is the air.
It is the, right, it is this miasma idea, right? But it's as he does, does this, does that, and then dips it in there, right? That's kind of showing this, right? So that seems, it seems like we've just shown this, right? And at the same time, right, we're already visualizing bacteria, we're talking about this stuff. We're already thinking about microbes, right? It's not, back with Reedy and we've never seen anything under the microscope, anything like that, all right? So we think this is all good, but I mentioned the broth. And there's still naysayers, right? So this broth that he cooks up is a meat infusion, all right? There are other bacteria, and I mentioned this to you guys, and we showed those spore-forming bacteria with the little green bits in there. Those spore-forming bacteria, like bacillus, they are all over the place, all right? And they, on stuff like vegetables or whatever, they are there. And if you cook it up and then cool it down, those spores that were formed, all of a sudden, they go back into the bacterial phase. They exfoliate, they hatch, if you will, and they go back and grow, all right? So another person thinks, I'm gonna repeat this, but instead of using meat, they do a vegetable broth or a hay infusion. They take this stuff, they cook it up, and now we've got a broth, but this is for the vegetarians. We don't want meat, we want vegetable broth, right? And so we cook this stuff up, and now we see it going turbid, all right? And we didn't do anything. It's the top one, right? The top one went turbid, right? And now we've got somebody that's like, wait a minute, right? It is spontaneous. Pasteur doesn't know what's going on, right? There's still things there, right? But like I said, with these bacillus that can form spores, this is the problem, right? So another researcher, we don't have him up here because Pasteur is definitely much more well-known, right? Thanks to, your carton of milk. Another researcher called Tyndall, I think it was Joseph Tyndall, Tyndall figures out a way to deal with this vegetable broth, all right? And this is also, as we're thinking about it, we're heating stuff up, we're boiling it with our Bunsen burner, we're bringing something up to a boil, but that's all we're doing, all right? Bacteria that can form spores, bringing something up to a boil isn't going to kill them, all right? It's going to kill some of them, but some of them feel that hot water and they are able to quickly form a spore and protect themselves. And then they hang out as the spore in the boiling hot water and it doesn't do anything to them, all right? Then that water cools off, goes back to room temperature, and now it's nice and hospitable. And there's also a boatload of nutrients in here, right? So these bacteria exfoliate or hatch out, start feeding, and make it turbid, all right? And Tyndall's like, wait a minute, I think I can figure this out. And this, like I said, or maybe I didn't complete my sentence. This is before we've got a pressure cooker or an autoclave. We don't have the ability to heat things up above boiling, all right? Once you bring a liquid up to a boil, it's 212 degrees or 100 degrees Celsius. It doesn't get hotter, right? Even if it's boiling really rapidly, that's what temperature it basically is, all right? So to kill these spores, they have to be at temperatures well above the boiling point, all right? And in order to achieve that, you have to put them under tremendous amounts of pressure in order to get that temperature up, all right? That's what we think about today, all right? But back then, we didn't have that technique. So Tyndall comes up with this idea, which is technically still used, but not very common, all right? Tyndallization is a process where you take a substance And maybe this substance is something that can't tolerate being put under intense heat and pressure, right? Maybe it's something that can't do that. So that's why we might still use it. Tyndall basically took this broth and heated and cooled repeatedly for about 5 days. He does this. And I think people aren't going to do any sort of home canning, food preservation that takes 5 days of cooking and cooling their beans to ensure that they're free of any sort of, botulism or whatever. All right. So basically, as you heat it up, some of those cells die and a good chunk of them form spores. You cool it down, the spores hatch out, you heat it back up, some of them die and a few of them form spores. Cool it down again or heat it and then cool it. And over the course of about a week or so, you get to the point that it just killed them all, right? There aren't any left. And then his vegetable broths looked just like Pasteur's in the flask at the top, nice and clear, breaking the top off, and they get turbid again, all right? So Tyndall basically did a slow cooking process of heating and cooling to basically get all of those bacteria that could form spores to basically die off over the course of time, all right? Kind of crazy, all right? And that was truly the last bit, all right? And shortly after that, we start seeing the spore-forming organisms and some of that stuff. Really pretty crazy, right? And like I said, it took a long time, a super long time, just like the miasma idea and thinking about this foul air. It took forever to get people on board that crap doesn't work, right? Even though you might smell something that smells super terrible, that doesn't mean you're going to die of some disease the next morning, right? It definitely does not, but that was what they thought, right? So we showed this before with Robert Hooke showing our first cells, a cross section of some of the cork, and getting the idea of thinking about cells, right? Virchow and Ramak, both of these guys really coming up with, or not coming up with, but popularizing the idea of the cell theory. So with the discrediting of spontaneous generation, we start thinking life comes from life, not spontaneously. And Virchow popularizes this theory with his cell pathology essay, and Ramak basically blows that up as he starts talking more about it, and it becomes kind of the main idea. All right, this is how we can describe where these cells or this turbid looking stuff shows up in the first place, right? Bacteria give us more bacteria, right? Or the fly lays the egg, right? Life from life, not spontaneously, right? And then, and we basically add in there that everything, that with the cell theory, everything is composed of at least a single cell, right? With the potential for it to replicate into numerous cells, if we're talking our bacteria, life from life. So we talked a little bit before about differences between prokaryotes and eukaryotes. And the prokaryotes really, they're like bare bones. They've got, nucleic acid and their shell, a plasma membrane and a cell wall. They've got the, enzymes and structures needed to acquire nutrients and to replicate, but they're not all partitioned off in separate regions. Eukaryotic cells are larger and partition these roles within the cell, and that's what we're looking at here with multiple things, right? So the endosymbiotic theory is an idea, a theory, but an idea as far as how mitochondria and chloroplasts came to be inside of eukaryotic cells, right? Both of these were types of bacteria, different bacteria, but types of bacteria, right? And an ancestral cell, a proto-eukaryote. So before it was truly a eukaryotic cell, maybe, bridging the gap, some sort of archaeal cell that had differentiated the littlest bit, possibly, picking up something else, all right? There's a handful of things that are showing up in here, more than just our endosymbiotic theory, all right? Which is happening down below. We can see things like plasma membrane or membranes surrounding structures like the nucleic acid and the formation of a nucleus. As we think about cellular reactions, there's all sorts of things that happen as we start degrading molecules and all sorts of things that we might think of as maybe toxic substances that might be being generated in this process. That could be very detrimental to the cell. And the last thing you want to mess up is your instructions, right? Your nucleic acids. So walling it off with a membrane helps to protect it. It gives it a little shield that makes it so any of these chemical reactions aren't detrimental to our instructions, right? So that gets walled off. There's more pushing inward, right? The cells are constrained by, we think about diffusion with nutrients coming in to these cells and the ability to get things spread around. You cannot be a cell that is gigantic, right? And think about really being very efficient, right? So even though eukaryotic cells are quite a bit bigger than bacterial cells, they are still really small, all right? And a lot of that has to do with how they can deal with energy, all right? So thinking a little bit about that, I like to envision this as we see all of this, maybe if we see it, as we see all of this material kind of showing up in here, the membrane around the nucleus, and then other endomembrane systems as we move through here, like the endoplasmic reticula, the rough, the smooth, Golgi apparatus is really all part of that. All of these things are different areas, different membrane-bound structures within the cell with different roles or functions, right? The bacteria are doing similar things, just not partitioning it all apart, all right? So as this cell gets maybe a little more proficient at feeding, now we've got maybe some more nutrients, we've got some more resources, we start thinking about trying to maximize some of these potentials. And I like to back it up. We're going to think about it in the cell here in a second, but I like to back it up and think about it from a person standpoint. And if it was you, you're all fine and good, right? One little cell, right? This is just you, right? And then maybe there's a significant other, right? And you decide, maybe we should move in together, right? So now there's two of you, right? And maybe you were in a little like studio apartment, but your significant other moves in with you, right? A little cramped, but if you can make that work, you're good to go, right? So then let's think maybe there's a bun in the oven, right? Studio's probably not going to be big enough for that little fresh hatchling, right? Maybe we need something bigger, right? So we could move to a bigger place. We could get a bigger shell to live within, right? Or maybe we could add on, we could expand. But the cell, it can't just get bigger and bigger and bigger because it's constrained by stuff like energy efficiency and things like that. Can we get all of these nutrients in? I can't just be gigantic just because I want all of this stuff, right? How can I deal with it? So instead of moving out and building on, we build in and we start pushing in the plasma membrane. We get all of these infoldings of membrane that basically increases the surface area. It increases the potential to do stuff on these membranes, but on the inside. And that's when we start partitioning different roles and functions within there, different regions on this membrane that has bunches of proteins that allow something to happen, or something else on another chunk of membrane that's all enfolded inside of itself. We've added extra rooms, right? Instead of building stuff out. We've built it in, all right? Those are all invaginations, infoldings of the membrane growing inward as we're trying to do more with our same space, right? So these are the precursors of eukaryotic cells with gaining this extra membranous material inside of them. And these suckers need to feed, right? So they engulf other stuff. And at the time, lots of these cells are in an environment that are very lacking in oxygen. We don't think about aerobic respiration like we think about today. It was very different, right? But there were some cells, some bacteria, some cyanobacteria that were starting to photosynthesize, were starting to produce oxygen. It's this kind of dawn of the oxygen revolution. And now as oxygen's accumulating, lots of cells are in a situation where this stuff is toxic, right? How do we deal with it? And there are some other bacterial cells that have evolved mechanisms that allow them to deal with the oxygen and to utilize it as part of their electron transport chain, and they can maximize the amount of energy that they achieve. With photosynthesis and oxygen being generated, aerobic respiration also shows up on the scene, and we start breaking down these molecules and liberating huge amounts of energy, right? And these little aerobic bacteria do great jobs of that. So this ancestral organism engulfs one of these aerobic bacteria, and now all of a sudden you've got a little oxygen sink inside of your cell that's able to take in that oxygen and use it and produce copious amounts of energy, right? So now this cell, instead of feeding on these guys, and I'm sure some of them were consumed and they ate them, but in the process of doing it, others that were inside achieved a huge amount of energy for that cell. And a symbiotic relationship is where both partners benefit. Endosymbiosis is where one is living inside of the other. And there are numerous other instances of endosymbiosis, right? But this guy takes up residence and ultimately becomes the mitochondria inside of the cell, a double-bagged structure where it was engulfed by one cell and that membrane surrounded it. The bacteria already had a membrane. Now we've got a double membrane, right? The same with the chloroplast, showing these suckers were eaten at some point, but they didn't get digested, right? And that's up here with our photosynthetic bacteria. This idea, we call this the primary endosymbiosis, and the second bit was the secondary. And we call it that because this happened after the mitochondria. Everything, thinking eukaryotes, not everything, but every eukaryotic organism has mitochondria in them, right? But not everything has chloroplasts, or we'd all be green, right? The chloroplasts were engulfed a little bit later on after the mitochondria, and that gives rise to plants, right? And we can see up here, mitochondria and chloroplasts, and below just the mitochondria. Really pretty cool stuff as we think about it. And with that said, a symbiote, you can't live without your symbiote. You can take the mitochondria out of a eukaryotic cell and you can watch both die, right? They can't live on their own, right? There are other ideas that help to solidify this as we look at the details of the DNA. Mitochondria have their own DNA, right? And their DNA is circular, like a bacterial chromosome, because these suckers were once bacteria, right? And they're inside of eukaryotic cells with linear chromosomes, right? As this process has evolved over the course of a huge amount of time, some of the mitochondria is picking up information, picking up proteins from the cell. It's helping it out, but the mitochondria builds almost everything that it needs, all right? All based off of its chromosome, right? A little bit is supplied to it from the cell, right? And again, that's why they're dependent upon one another, right? And the cell needs it for energy. Okay, this next bit, there is actually a YouTube video. I'll try to find that and just post it. I'm not going to show it in here, but really pretty interesting. Ignaz Semmelweis was a huge proponent of hand washing. He kind of, it almost destroyed his career, really, right, as we think about some of this stuff. So he recognized some issues, right, and he tries to, express this, and it's almost like we're flipping it back to spontaneous generation, right? There are lots of people, lots of guys that are like, this is nonsense. Don't tell me what to do, all right? Basically, at the time, physicians, they're all male, right? So you've got all of these doctors, guys that are pretty sure they know what the hell they're doing, right? And I don't need some other guy to come in here and tell me what I'm doing and that I should maybe change what I'm doing, right? I don't need that, right? So Semmelweis is at this hospital, and it's a teaching hospital. There are lots of students that are learning to become physicians. They're working on cadavers, they're working on dead bodies, and they're also delivering babies, all right? And there is also a wing in this hospital that is more like the maternity ward. There is a wing that is full of midwives. The midwives are all female, right? And they are delivering babies as well. And Semmelweis recognizes that this side of the hospital, the doctors, have a huge issue with women coming in to deliver babies, and they never leave, right? They get in, they get sick, they die, and the baby dies, and we've got huge mortality rates, all right? You almost just kind of know in the back of your mind, if you're going in to deliver a baby, it's like 50-50 chance, right? I may not make it out of there, right? The video shows stuff like, the ladies, before they would go to deliver, they would pack their bags so that, you're ready, you got some stuff in there, but they would also get their death clothing out and they would get all of that prepared. And that just creeps me out. I'm like, holy crap. Here you are thinking of bringing this little bundle of joy into the world. And in the back of your mind, you're like, I got to get prepared for when they bury me because I'm probably not coming out of this, right? Makes you kind of think, shit, man, do I even want to have a kid, right? What's going on? But you can't think about stuff like that, right? So doctor side, lots of death. Midwife side, hardly none, right? And he wonders, what in the heck is going on here? How is it that this is occurring, all right? The midwives aren't working on cadavers. They're not dissecting dead bodies and trying to understand stuff like that. That's not what they're doing, right? The other guys are, right? Midwives are also maybe a little more conscious of what's up. And as this lady comes in to deliver, they wash their hands, right? They prepare fresh linens. They do all of these things to keep things more clean. And the guys learning to be doctors don't give a crap about that. They're working on the dead body. They turn around, reach in and deliver a baby, and they go back to the dead body. They don't wash their hands. They don't care because they're not spreading disease, right? But they were, right? So he starts saying, you ought to wash your stinking hands, right? And all of these other guys are like, no, right? And over the course of time, it becomes common practice, right? To wash your hands. But there were numerous people trying to discredit him, numerous physicians trying to discredit him because he didn't know what he was talking about, right? But ultimately, we recognize, yes, he did, right? Lister, Joseph Lister, he came up with, and this is right around the same time, ideas with disinfectants, Listerine, right? disinfectants and care of surgical wounds, sterilizing equipment, all of these things, along with hand washing procedures, helps to dramatically minimize the infection that was being spread just by going into the hospital, right? Crazy. Koch came up with his protocols, as we, I think, mentioned, the Koch's postulates to look at the spread of disease. We see this infection, we grow it up, we infect something else, and we're able to show this is what caused this disease. He did all sorts of other things, right? But we're all, at this point, starting to progress and think about what we're doing, right? And microbes are really behind this. You know, I don't think we've got Florence Nightingale in this mix, but we should. She also was a huge factor in this. And as we see, you know, I can't remember exactly where we were at, some battlefield, the Army hospital, basically the people coming in with all of these battle wounds, gunshot wounds, and all sorts of massive damage. You'd think that's what's killing you, but no, it's coming in and you're in a bed that's all, you know, full of soiled linens. There's sewage on the floor. There's all sorts of issues, problems, right? And as she shows up, she's like, we ought to clean this stuff up, right? There are numerous things that she did right along with all of this, thinking about like aseptic technique, steps that we're trying to kind of introduce to you guys in the lab to begin with. All right. So shifting over the littlest bit, but we could still be thinking Robert Koch because he was fundamental in evaluating stuff like tuberculosis and coming up with the acid-fast staining procedures that allows us to see those bacteria in the first place. Here we've got two images, normal lungs and then the infiltrate suggestive of pneumonia. And as we mentioned, lots of things that can cause pneumonia, not just tuberculosis, right? But I think that's really what we are seeing there. So kind of a timeline as we move through here. We've got the Greeks with the miasma theory, where they're proposing this long ago. And then we start progressing, trying to discredit spontaneous generation. As I said, it took a super long time, centuries, right? As we were moving through this, right? And really the 1800s were rich with discovery. John Snow with cholera, Pasteur, Koch, all of these things, 1800s as we're moving through here. And here is our pasteurization a little bit before the spontaneous generation. So pretty interesting. This shifting now, we want to think about cells, bacteria versus eukaryotic. And this cell, we'll quickly talk about it and then stop. This is a simplification, maybe a generalization. There are lots of things shown up here, and there are some bacteria that would likely have all of everything we're showing you, all right? There are others that we kind of can mix and match a little of this, all right, as far as what we see. We're looking at one bacteria that's a rod shape or a bacillus shape. Remember, there are different shapes of bacteria as well. Typically, when we think about these, there are potentials, not all of them, but lots of them can have flagella. We'll see some images next time, but there are some different ways that the flagella will be oriented on the cell. We can differentiate some of that stuff. We're not going to get super bogged down with all of those details, but I can pretty much guarantee there's a test question about the arrangement of flagella, right? Which end, all over, whatever, all right? And like I said, we'll hit that next time. So I started with that. We can see some of them have a flagella. There are little hair-like projections, little fimbriae or pili. Here's a pilus that's a little bit longer. The pilus helps for bacteria to bacteria communication. We'll touch a little bit on that later on. The fimbriae are kind of almost like Velcro that helps cells to stick to something else, maybe another cell, maybe a bad guy getting into your body and sticking to some of your own cells. The fimbriae kind of help with that. Some of them have an outer bit that's a sugar layer, right? A capsule layer, not all of them, all right? Some that are pathogens, the bad guys, we really think of them as one of the good mechanisms for them to be a bad guy is having a sugar coating because it helps to prevent our body from seeing it, right? So some have capsules. We can see the cell wall as I move up here, but we're peeling it back. Cell wall, virtually all bacteria have cell walls. The ideas behind it, size is a little bit different. And then a plasma membrane, right? Inside of it, all of the little red speckles are ribosomes free floating in the cytoplasm of that cell where protein synthesis is going to occur. The chromosome, circular, single circular chromosome, kind of in the center of the cell. We say that region is the nucleoid, but it's not a nucleus. It's just kind of where we sort of expect to find it, right? And I don't really get super bogged down with that. There are possibilities of some inclusions, and when we showed that spore stain, I mentioned sometimes you might gram stain something up and see little hollow bits, and the question should be, is it a spore or is it an inclusion, all right? You might see that, and then spore staining would help you to differentiate. Down below is a plasmid. Some bacteria have it, some don't. It's extra chromosomal material. It helps to impart things, advantages, like maybe antibiotic resistance or toxin productions or something like that, something that it doesn't have just in its regular chromosome. And they possibly can share this plasmid through its pilus to another bacterium. And I think with that, pretty sure I covered all of the anatomy of that cell. We will quit with that. I will see you all Friday.